Okay, hello everyone. Welcome to this week 9 lecture. We're going to start because we've got lots to cover today, as with last week. I'm trying to include more of the asynchronous in the synchronous, just because I see that this is more interactive. We see a bit more engagement, and I'm not sure if everyone has a chance to cover the asynchronous. Yes? Yeah.
Can we access the slides, like this one, during the lecture?
Oh, the PowerPoint slides.
Yeah.
Okay, because I make last-minute tweets even until the beginning of the lecture, that's why I haven't done it. But what I can do is I can upload it from the next session, so you guys have it ahead of time. Yes. But for this one, I could do it. But since we've already started it, let's start it from the next time. Thank you. So this week we're talking about the excretory system. So far we've only discussed anatomy. We're going to talk a little bit about diseases, but not as extensively as the cardiovascular system, because we're going to put more emphasis on these devices, the medical innovations in extracorporeal life support systems. And this is really important because this is the summit of engineering in biomedical research and biomedical sciences, is to be able to produce a system that can replicate human organs, okay, with all of its sophistication, okay? And this is interesting because, as you'll see throughout today's lecture, it didn't start in the past 10 or 20 or 100 years. It's been there for a long while, even before we had knowledge of tissue engineering. So even before we knew how tissues behave, engineers were brave enough, and clinicians were also brave enough, being surgeons, they actually to use these systems without having enough proof and enough evidence as to their performance. So when you look at the timeline as we go through the slides of this lecture, you'll see how starkly different it is from today's development phases. Today we're going really accurate, but not as fast, not as risky. But of course, we're not trying to risk anyone's life, so that's why we've gotten a little bit more careful about this. So the image you see here is a lung transplant model, and although this is a 3D model, but it's very much the system that they use currently in United Therapeutics. So we'll discuss this, but today's focus, as this slide is suggesting, is organ engineering, organ transplantation, xenotransplantation, organ production, manufacturing, and all of that. So we're taking a quick detour from our regular course to talk about these topics. So this week we're talking about biomedical devices, implants, and innovations in the excretory system, as I mentioned. And we're going to go back to diseases in the asynchronous stuff that I'll upload for next week, but for now we're just focusing on devices. So this is an actual beating, expanding lung, okay, right before they're going to transplant it into the other human, the recipient, okay? It's not just the air that you pump in, it's also the blood flow. It's the temperature of the blood that you have to regulate. It's the hydrodynamic conditions of the surrounding fluid that produces the osmosis. It's a lot of those variables that you have to make sure that you tweak in order to keep it alive. And doing that on the motion is an even bigger challenge. So organ transplantation so far, if you've got 10 organs, probably 8 of them will make it to the site, to the recipient. 6 of them will be fine throughout the surgery, and then you may have just 2 or 3 that survive a long while, okay? And a long while, I mean a year. Really, the problem is they don't even survive beyond that often. You may know this guy if you're a Formula 1 fan, Nicky Lauda. He was a 3-time world champion, and he actually had a major crash. That wasn't the reason he actually got a lung transplantation, but it may have induced those respiratory issues for him later ages. But then, look at this. He got a transplant in 2018, August, and then, unfortunately, he had a history of these. So he had his first kidney transplant in 1997 from his brother, and then in 2005, the other kidney from his wife, and then, unfortunately, he passed away only 8 months after he got the transplant. Okay, they're not sure if it was the lung, but it was actually on a kidney dialysis system that he passed away, meaning that he needed so many frequent kidney dialysis procedures that the kidney just wasn't taking it. What's going on? It's not the lung that wasn't performing right, it's the load of the lung that doesn't belong to that body that the other organs have to take, the kidney, the liver. So these other organs have to take that extra load coming from all the excreted species, all the ionic exchanges that are not supposed to be there. Filtering that, the kidney has to work double load. Okay, so and you know already what the function of the kidney is. Okay, it gets rid of all those toxins. There's a huge exchange site there with these intricate structures that we've already sort of discussed briefly. But that's why it fails. So it actually isn't the optimal solution, even if we had enough donors, like we have in the UK, fortunately, based on the statistics. It still isn't going to be enough because there's a lot of loss. We have enough donors, but it doesn't make it to the recipient. And even if it does, it's rejected. Now, it's interesting. The lists in the UK report that there is enough donors, but that's actually just to meet the supercritical patients that really need it or they will die. There are probably 10 times more a population that need an organ, but just aren't considered eligible, right? So we aren't actually that close either. So it's neither that we have enough organs, nor that they make it through and can then sustain their performance. So that's why, from a long while ago, even before they did any of this, this is about 400 years ago, right? They were thinking about a replacement. And this replacement, the best option seemed to be animals. Xenotransplantation, okay? Because we had an abundance of these animals, and the idea was, I mean, it started back here. You can see they're trying to fuse the human circulation with blood from a tiny goat or a sheep, okay? And this was actually successful the first time that Dennis actually tried this 400 years ago. It was successful in a kid, 15-year-old. It worked very well, and he survived, and everything was fine. And then he repeated it twice, a couple years after, on adults. And this time, it killed both adults, okay? Of course, the kid probably didn't see a severe reaction, an allergic reaction, because the amount of blood was little. But that then allowed him to continue on, and then unfortunately he had 2 patients that died. Of course, now it's obvious to you guys why we can't just put blood from an animal into a human, but back then it was a big thing. So it's been a concept out there for a long while, until in the 1990s, this lab actually figured out that there's a specific sugar that activates the immune response, and if we could knock out the genes that are responsible for secreting that sugar, we could actually down-regulate the response of the immune system. And it actually worked very nice, and that was why Novartis, who's now out of business unfortunately, a majority, spent a huge deal of money trying to make a farm to grow pigs with very high standards to actually meet that xenotransplantation milestone. But then it actually didn't die there, okay? Xenotransplantation continued on. And January this year, less than a year ago, we saw that United Therapeutics announced their first xenotransplanted kidney from a pig into a human that actually survived. So in terms of the metabolic rate, in terms of its performance, in terms of longevity, so far it's almost a year. Patient's doing very well. It's actually an athlete and he's doing pretty good. So this was the first example, but then they have a couple other xeno models in the pipeline, the heart and the lung, which is right now also sort of proprietary development. They haven't announced much. But they are not just developing the organ, as we'll talk about that. We'll talk about how they're printing and producing these models, but they're producing the entire ecosystem around it. Okay, so it's the drones that then transport the lung to the patient. It's the perfusion system there that's very complicated and has to produce that pulsatile nature of the cardiovascular flow. It's all of that. So this company is putting a lot of money in developing all of that pipeline. Now, away from animal-driven stuff, let's talk about what we can engineer today. Okay? Today, we have extracorporeal life support systems. And what are these? It's any object outside the body that takes the function of a human organ. Okay? It can be a lung, it can be a heart. And today we're going to talk about oxygenators, which are a very important class of these extracorporeal systems. And as you'll see, the main problems with these is because the engineered surfaces are not as good and you get blood clotting and thrombosis, you have to use anticoagulants. And anticoagulants means you don't get coagulation, right? That means if you bleed, you don't get wound healing as well, right? So that's the big issue with these, is that these patients, if they have a tiny bit of a peel on their skin, they'll bleed so long, okay, that there has to be a medic's care immediately, and they have to use some sort of sealing mechanism, a biological one, to actually seal the wound. So there's a downside to it. They don't suggest it, they don't actually advise it as much in the elderly, which are the group that actually need it, because if they bleed, their system is even weaker, so they can't really manage to deal with that. So what are the other issues? I mean, blood clotting disorders, okay? You're shifting all these hormonal concentrations. You shouldn't expect to get the same behavior, so you get clotting in places that you don't want it. And then there's infection, because what's happening, there's something outside of the body that's connected with the tubing to the inside. Keeping that sterile is a big business. A lot of the companies are just formed around designing sterile patches that can seal the tubing when it's entering the body without bacteria passing through. So this is really big. And then sometimes, if the pressure profile in that system that circulates blood isn't set properly, it won't reach all the way to the legs and the arms, okay? So you can have basically that same process that happens in patients when they're in a hospital for a long while, they're just keeping themselves horizontal. It happens here. It's called ischemic reaction. So that's one, and then you've got stroke. Okay, so you've got stroke, heart strokes, because the heart isn't used to that beat. when you're priming it at the beginning of the process, you're inputting this load on it and it can't take it. So sometimes you have this resonant effect, and this actually makes the left ventricle go out of sync for a while, and that can quickly basically expand and accelerate in its process of affecting the entire circulation. So when do we use these systems, these oxygenators? It's both used for the lung and the heart, and you'll see why in a second. It's not just for the lung. It has the function of the lung, meaning that it oxygenates blood that's being circulated through it, but it's also used for the heart replacements. And usually it's these diseases, but only if they're super severe. So blockage in your pulmonary artery is something you can actually eliminate usually. But in some patients, look at this. The entire pulmonary artery path has suddenly clotted. Now you may think with yourself, how long did it take to have this clotting, right? If it was just in this tiny region and was blocking the flow, the patient should have figured out and they should have gone into emergency. How did it get enough time to clot this entire thing? Well, the thing is, it happens in a very quick period. Blood is a viscoelastic material. As shear changes, it's like you've got cornstarch and you're punching it, right? It's shear thickening. And as soon as you have one region clogging, it can propagate. But also, these markers and molecules that initiate this clotting are available everywhere in the flow. So suddenly, when the homeostasis doesn't stay stable, everything goes south. So if this happens, that lung is no longer functional. Now this, the surgeon has successfully removed, which is a big deal, right? They've been able to go in and extract it. But in a lot of the cases, it's so broad that you cannot do any sort of treatment apart from replacing the entire lung, okay? COVID was an example where, especially in China, they used a lot of transplantation to solve that problem when it got so severe that the patient was dying, basically. There are congenital diseases, okay, diaphragm, so in fetus lungs, basically, when they inhale the waste product that they shouldn't be inhaling, it quickly deteriorates the tissue and causes a malformation and malprogression of the tissue structure, and so it has to immediately be replaced as soon as the mother has given birth. They have to quickly replace the lung. Sometimes it's too late. So, and then there's heart attack. That you have various sorts of heart conditions that also the heart is completely shut off. There's no way to deal with it. If you have a little bit of time, you want to connect it to ECMO. Now, what is ECMO? Extracorporeal membrane oxygenation. These systems, now from an engineering perspective, have progressed quite a lot since a long while ago. It's about 80 years ago that it started, and the idea is that you draw blood from, so there are two styles as I'll show in a minute, but usually it's either from the vein to the vein or vein to the artery, and what happens is you have to pump it because now it's coming outside of the body. Hearts are either involved or not. If they're not, of course you need to pump it. Even if they are, it's actually facing all this extra resistance, so you still need to pump it. And then you have the membrane system, okay? This membrane system up there is what actually is now a membrane. It used to be a different thing, as we'll look at, but it's what the blood gets circulated through and travels through to then be put back into the body. Now, these numbers are quite important. The surface area, remember that number, okay? It's about a few meters squared, just 1 to 2 meters square is what they typically have. And also remember the rate, the ventilation rate, okay, which is a few liters per minute. Now, these are the two styles, okay? What do you think the difference is? So one of them takes it from the vein and puts it back into the right atrium. The other one takes it from the vein and puts it in the artery. Okay? What do you think the differences are between these two? This is from the vein, after it's gone through the organs, it takes it, oxygenates it, puts it in the right atrium. The other one, it takes it after it's deoxygenated, after the organs, and puts it back before the organs. Yep.
The second one oxygenates the whole body, while the first one oxygenates just one organ.
So the whole body is down here, okay? So basically this is before the vasculature reaches the other organs, okay? This schematic isn't really accurate, but it's basically showing this is before the organs, that's after. Yeah?
Which on the right, the faster rate of oxygenation?
Why? What do you think?
Because it enters, so the oxygenated blood enters the heart.
Enters the heart.
Faster than the one.
So it is a consequence of that, but you just pointed at something important. It enters the heart, whereas that one doesn't. This one is in parallel to the heart and lung. It completely bypasses the heart and lung. None of them are used. In this case, you're still using the heart. So you're putting it into the heart's right atrium, and the heart does the pumping action. You're just oxygenating it, right? In that one, you're completely parallel to the system. You take it after the organs and put it back right before the organs. So no heart or lung is involved in that process, and that's when both of the organs have failed or the heart has failed, right? So these two serve different purposes depending on whether you've got both organs failing, the heart failing or the lung failing. You can choose one of these. And there you have a cannula. You can see how far it's extending here. It goes beyond the first branching, and here it's dipping right down into the atrium, and they have to administer this really fast. They quickly make an incision, put it in, and then prime it really fast. So the other challenge with this is the priming. If this circuitry is air-filled and the blood enters, all that air is going to be bubbles going back in the bloodstream. So you actually want to prime it with the full volume to fill those up. So that's why they take a large sample from the patient's blood right before to fill the circuitry, and then they actually use the circulation to go forward. So now looking at these devices, from the old day, they thought of a very clever solution. Why don't we just bubble it, right? Why don't we just inject oxygen, just like the aquarium bubbling systems, right? Why don't we just bubble through and get the blood oxygenated with that? Now the problem with this was there was no degassing mechanism, okay? But still, these were developed about 80 years ago, right? And they saved lives, especially, and this was sort of first rejected by the Minnesota hospital, but then this brave guy, actually, I forgot his name was Dewar, yeah, I think Dewar. So he was the first person who was brave enough to give it a go in an open heart surgery. And the good thing was open heart surgery takes about a few hours, less than 6 hours, less than 5 hours, usually just an hour or so. And then you won't see any effects, prolonged effects. It's used, it's taken away, it saves the life during the process, and that's it. So it was quite successful. It came back and it was used around the 1980s, and then it was kicked out because there was a better idea. Now this better idea was these oxygen flow system that basically ran oxygen over a film, and what happened was you had gas on one side and the blood on the other side, and the blood was still in direct contact with the air, so through those pores of the membrane, and it actually got circulated. But the problem was that it's just the volume, the surface to volume ratio is so poor that it didn't actually get enough oxygen, and it didn't actually circulate much volume if you did want to do that. So it was replaced with these more sophisticated versions, which still had a porous membrane that would serve as the oxygen exchange surface, and then they would actually wrap it into these tubes, and they would sort of have oxygen running in one concentric tube and the blood in the other, so it was sort of separated, these layers of blood, oxygen, and so it was a little bit more efficient, but still nothing like the human circulation, right? So The most recent versions, they use these micro fibers, okay, these very thin hollow fibers that are only a few tens to hundreds of microns in diameter. And they actually have blood flowing through these meshes of fibers and air and water traveling through the other two. And what happens is, why do you need water? To regulate the temperature of the blood, right? So that's what it does. It's like a heat exchanger. At the same time, you have gas flow, oxygenating the blood. And what's the problem with this? I mean, it looks brilliant, and it still is the best system we have. But based on what we've learned from cardiovascular, what's the problem with this? Huh?
Time?
Time. So you mean, what do you mean?
How long it takes to...
Yes, so it doesn't get oxygenated as good as we want. So if we really want to replicate that, we need a much larger construct so it has more residence time. So actually we're not slowing it down the way the bloodstream does. But what can you say in terms of, if we, okay, let's say if we make if we have the ability to make these at the single micron level and run blood through it, okay, super fine mesh, and we just make a large construct like this large. What's the problem with having all this structure at the capillary level? Remember, it doesn't have the arteries, arterioles. It's all capillaries. What's the problem if our body had capillaries starting from the heart all the way through? What was the issue with capillaries? Huh?
Surface area.
Surface, well, they're efficient in terms of surface area, and we have them at the organ level, but why don't we have it all the way from the heart? Yeah.
Because the velocity of the blood flow is quite slow at capillaries.
The blood flow is slow, yes, so we need it to circulate faster, but what else? Yep.
High resistance.
Resistance is really high too, so those are the issues. You need a huge amount of pressure from the heart in order to be able to circulate if it's all capillaries. It's so high in resistance. So that's the issue with these. You have a pump there to support that issue, but what happens? The pump is squeezing it with such high pressures, the shear on the cells induces coagulation and clotting. And inside these meshes, you start getting blood clots and thrombosis. And then the device usually is coated with something that makes it clotting, but that's even worse, because then it can go with the flow, go in the patient's body, and then disaster can happen right there. Okay? So, although these don't fail that fast, they stay there for 5 months, 6 months, but they actually can cause issues that are fatal. So that's why you've got to be really careful in designing the flow rate, the pressure profile, and all of that. And the typical material, we'll look at the material candidates, but is polymethylpantene. And what you actually have here is the fibers that are aligned very well. So there's a brilliant process to manufacture these. They can also electrospin them on large surfaces. And these are on the sort of wider end of things. They're about 200 to 300 microns wide, but you have fibers now around 180 microns. And so, as you can see, the resistance there to get blood through this gap is going to be super high. That's why you need to pump it really strongly, especially with the thick blood, with all that sort of viscosity. Now, back to one of the markers I mentioned. Gas exchange surface area. The best that Mackett can do, which is one of the best oxygenators in the market, okay, is only about 0.8 square meters. Well, they've done recent advances to get it to about 2 to 3. And you see all the other competitors also in the same range. So it's about 1.2, 0.7 meters squared. Okay? How large of a surface area do you think our lungs have? Very high. Very high. How do you think it's in the order of a meter square? 5 meters square, 10? 30 meters square?
I read that, isn't it, you can extend your lungs out
So yeah, that's the length of the capillaries there. Exactly. That's actually a big marker too. But I don't think it's quite that high. I think it's basically about 5,000, 4,000 miles. Okay, so it's about the width of the US. But the surface area is interesting. So, and note that the materials now they've all converted on polymethyl pentane. I'll talk about the issues of that in the biomedical materials lecture asynchronously. But this is the size of the human lung surface area, right? It's about 100 square meters, and we're at 1/100th of that, or let's say 1/30th of that. So you're putting so much pressure in it, you get coagulation and clotting, you don't have the surface area, the exchange isn't as efficient. Why do we stick with it? Well, the patient will die otherwise, right? So we're keeping them alive. Even for an extra day, they might be on the transplant list, they may get their transplant, okay? There may be some signs of recovery of the system, but it's really not there yet, right? So oxygenators, not the perfect solution so far, but they are working, they are saving lives, and they save quite a lot of lives. But these programs, these extracorporeal life support programs in any country are very rare, and they need a very significant amount of funding. They need about $100 million to be able to support 100 patients in the process of a year. And that's, that's one of the larger systems. But the interesting thing is they actually charge the US healthcare system $1 billion, so they get 9x return. So the investors who make these hospitals actually get 9x return. That's the part you don't see. So they're really like, we don't want more employees, we don't want more, systems. But it actually makes a lot of sense to do that. But it's a very advanced technology that not all hospitals can actually afford to initiate, but they need to find the investors, just like any other business. Anyway, so that's why United Therapeutics needs to 3D print these vascularized tissues. Or the alternative is xenotransplantation, but taking all the cells out. We call that decellularization. So we wash all the cells out, and then we recellularize it with the human cells. Okay, so either of those two. But you really can't work with membrane oxygenators if you want to implant it in the body. So how do they make them? So you already know about 3D printing. This is the process they use to make them, okay? And I know that because I've been at their site and we've actually delivered printers to them to do this task. The way they do it is with these photoresins. But what's the challenge here? Let's say you want to make a lung model, okay? So this is sort of an alveoli model. You may have seen this. How many of you saw this before from my previous presentation? Okay, wonderful. So what it does is it beats, it gets oxygenated, the blood that passes through from the inlet to the outlet. And this was the printer that we sort of manufactured and sold about 300 of those. And it's interesting, actually, it's not that sophisticated, but it costs the price of a Tesla, about 30K. But I think we're marking it up a little bit, but that was because we needed the funding to develop these organ programs. But honestly, there was also the various other aspects to this that made it a product that people would sell. Otherwise, it's not a charity. People don't just invest in you. But the problem is, you need about, not 1,000 of those, 400 million of those to build a structure. And each of these alveoli sacs is supposed to be about a hair in thickness, and the channels are supposed to be 1/10 of a hair in thickness. And the model that we've made there is the size of a coin, right? So improvement-wise, we need to have, if you translate, so I'm going to actually go back. What's the shortcoming here in terms of the printers that we currently have that use light? Why can't we do it with the printer that we use in the lab? Yep.
It's not as detailed.
Not as detailed.
Small prototype.
Okay, so what defines the detail in a light-based system? Yep.
There's too many layers for the light duration to be as detailed
So layers, exactly. So you want to have super fine layers, so 1 micron layers rather than the 50 micron which you guys used in the lab. But what else? So apart from the z direction, in the sort of plane, what defines how small the objects are? So there was a screen there, a display, right, that was projecting the light. The pixel size of that display, right? How small the pixels are. And in projector space, in LCD or LED space, what's the best pixel count that we can get now? 4K displays, 8K displays, right? So it's about 4 million, right? 8 million pixels. We actually need about that many voxels. Now voxels is a different unit. It's not pixels, because pixels is in the plane. Voxels is in the volume. Okay, because now you've got the layers, as you mentioned, okay, so you multiply by that, then you've got tiny cubes that build the volume. We are at a point where we can only produce a gigavoxel, and we actually need teravoxels in order to be able to produce the sophistication of a 5 micron, 10 micron capillary at the volume of a human lung. So the problem is that we don't have enough pixels, we don't have enough voxels, and even if we do, it takes a long while, right? Because processes are usually parallel, okay? They're layer by layer. So this is what we developed to actually solve that problem. And the idea is we've got an 8K image that was sweeping on the surface to produce what we call a 4400K image, okay? So if you look at those patterns, you'll see the tiniest features are about 2 to 3 microns, and the area of this is enough to basically cover one lung, okay? And if you then project this image onto that same system that you have and do it layer by layer, and you have thin enough layers, you engineer the chemistry so that it absorbs light just about a micron layer, you can actually engineer a full-size organ that can be produced with this. And that's what they've actually done, and I'll talk a little bit more about this in the next lecture. But it's the one thing you guys should know, and probably not many know, is that it's super slow. The reason it's slow is that you're doing it in a layer-by-layer fashion. There's a limit on how much energy you can transfer to the material, right? The cells die, okay? And if you're doing it one layer at a time, stopping, then going to the next layer, it just means that if you've got an object that's about 20 centimeters tall, and you want 5 micron layers, how many layers is that? So 20 centimeters, let's say just 10 micron layers. So divide 20 centimeters by 10 microns. How many layers is that? Anyone got that? So 20 centimeters or 20,000, 200 millimeters or 200,000 microns divided by 10 microns, that should give you 20,000 layers. 20,000 layers, and each layer takes about a second. Especially now that you're sweeping it, you need to even spend more time because each piece should get enough time. So it took us about a week to print an object this large. Okay, and that's not quick enough. And we did that not at 10 micron, but actually 50 micron layers. The pixel was actually 5 microns. So I'll talk more about this, and we'll talk about other systems to replace our kidneys and livers. But I mean, we've got about 10-20 minutes remaining. There's an important thing about this module, and that's you guys are the first to take this module. This is the first time we've delivered this module. It's also the first time we've put a lab component to it, which is why we had that first session, those of you who were there, was an utter mess because there were so many things that could go wrong with printers. Of course, that's the nature of them, but also the module itself. The lab component was too complex to say. But what we care about is to deliver to you something that you guys will benefit from in your future careers, and the skill sets that you actually need, the engineering intuition that can help you, not just a bunch of stuff that you memorize or a lot of formula. If you know just that 1 or 2 or 3 bits of formulation, bits of equation that you actually came across so far, that will be enough to take away from this lecture. It's not the volume of these materials that matters, it's the quality at which you understand that everything is simple equations, okay? So what really helps us is if you guys actually echo your voice, okay? Tell us about what you found good in terms of the practices, in terms of the module, and what you thought could be improved, okay? Tell us what you enjoyed and what you didn't. And I know that finding this time in your schedule is a challenging, tricky task, but since we've got a 10-minute time remaining to the end of the lecture, and that's really as important to the development of this module as is to anything else, I'd like to ask you to actually use this 10-minute slot to go on this. You might have received an email, you must have, or there's an ARCS website, okay, which you can find online. Log in there and just provide your feedback right now, because that's really something we've been missing. Without your voice, there are no solutions and no improvements, okay? Yep. Yeah. Final exam will be both dental materials and the soft tissue.
It's about as much dental materials as when we've got.
Yeah, the dental was a lot of material. That's why we've sort of reduced the rest of the load. But in the exam, it's going to be 40, 45 to 60. Okay, so that's the ratio of the questions. So it's going to be 40%, 45% dental, and the rest of the stuff you've covered here. But yeah, you guys should be totally fine as long as you've just got the lecture materials, at least for this 60%. Yeah. And the second quiz, I'll announce to you how we're going to do that, what the format is, but it's just about the stuff that you've seen in the lab. Nothing complicated, as long as you've just been there, experienced that, and just briefly listened to the comments I had, should be able to easily respond to those. It's really an attendance marker for the lab sessions. That's it. Okay, and I really yeah, go ahead.
The final exam is surely multiple choice.
Huh? It's multiple choice. Yes, it's multiple. It's about 50. Yeah.
Is it 2 hours?
Huh?
Will it be 2 hours that we get, or 1 hour?
You'll get the full, I think it's 2 hours, yes, or 1.5, and then I've got to check, but I'll let you know.
Is it one of those where we hour or time frame?
So you will be given a time frame, and then during that time frame you can start, as soon as you start you've only got that time to finish. We'll have to, we'll still actually, we've contacted the heads of education to see what the system has changed a lot, but it's probably going to be that the window's going to be open for a while and you can join. Yeah, that part I'll tell you about, but I'm not 100% sure because we're waiting for their response. The exams need to go through scrutiny, then they need to be secondary approved by another exam. So it's all of that process, and then we'll get these. But we'll get them finalised for you guys probably next week or the week 10 lecture. Yeah. Yes, so that, everyone, there's a question about quiz number 2, right? So quiz number 2, as I mentioned, is about the stuff in the lab, okay? And I'm aiming to have the window open for the entire day of that, basically, lab. And you guys, when you're in the lab, you can spend that 20 minutes at the end of your lab session. And it's a timed attempt, but you will basically be able to take it when your lab session is. If that doesn't happen, we'll have it in week 13, so you'll have a little bit more time to work on it. But then in week 13, it will be an online attempt, just like the first quiz, and you basically need the lab stuff only to be able to respond to that. None of the lab stuff is going to be in the final exam also, so it's just the material covered here. And I'll let you know what the final exam opening time is and how long you have to respond in the next lecture. Oh, yes. No, so each group that comes now, they have the window will be open from the beginning to the end. You'll have 20 minutes to respond. But I'll tell you that this is your 20 minutes to do that. You'll have the end of the session. But I'll let you know before we start whether it's going to be that or the final exam or the first three. Yes, we'll make sure you have enough. Is there one question? 2 minutes. Okay, 1.5 minutes. I'll discuss that with you guys, or we'll have a quick survey. Yeah, we won't. Yeah, we'll make sure that you guys have a fair amount of time. Okay, guys, well, I want to thank you for taking that time to complete the survey. It really helps us in developing the program. And that's it for today. Yeah, thanks a lot. Thank you.
Wait, what time is it? 9? 7 in the morning. Oh, wait. Vanilla, then what was it? Ingredient from the it was something really so it was vanilla. Yeah, I thought that was fun. So that's probably not true. I said it when I was like, what the hell?
What did you see in the corner?
Because we didn't do it last week.